Hi, my name is Sean Thornton, and I am a senior biology major at UNCG. I have been studying a gene essential for meiotic chromosome segregation in the fruit fly for the past year. So now let's talk about some more background information about chromosomes and their segregation. In humans, the normal amount of chromosomal pairs should be 23, which is going to be 46 chromosomes. However, if chromosomes segregate abnormally, genetic syndromes can arise. In Klinefelter syndrome, an extra X chromosome is in the 23rd pair. We can use flies as a model organism to study chromosomal segregation because they only have 4 pairs of chromosomes. It's much easier to track these chromosomes and study. So as we talk about chromosome segregation, it's important to go over how that relates to meiosis. So sister chromatids are identical copies of DNA, and there are 2 of them. So after meiosis 1 here, we see there's one sister chromatid on the left and one on the right. So these sister chromatids are going to stay together after meiosis 1. This is called monoorientation. They both go the same direction. In meiosis 2, that is when the sister chromatids are supposed to separate, and we see that they do separate in this diagram. This happens for the blue ones as well. In meiosis, it's very important that the sister chromatids monoorient in meiosis 1. However, the mechanism that this happens is not clear in many organisms. This has been discovered in yeast, and the proteins used in yeast are not the same as the ones used in the fruit fly. So I believe that I discovered a gene that is essential for keeping the chromatids together during meiosis 1 to make sure they segregate at the right phase in meiosis 2. Here are the results for the male meiotic non-disjunction experiments. And we see that when CG30383 was mutated, there was 4th chromosome misbehavior, and there was also XY chromosome misbehavior. Now, the 4th chromosome is an autosome, and the XY chromosome is a sex chromosome. And the frequency of this non-disjunction was random, around 50%. And this is the expected genetics frequency for a misbehavior happening at meiosis 1. The results of the female meiotic non-disjunction experiments also showed that there was misbehavior of the X chromosome and the 4th chromosome. The non-disjunction frequencies were also random, and these further support the expected genetics phenotype for a defect in monoorientation. This is a meiosis 1 dividing cyst that was captured on a confocal microscope, and the red color indicates an X chromatid, while the green color indicates a Y chromatid. During this experiment, we visualized that there was 70% non-disjunction of the sister chromatids. This is important because monopollen has very similar frequencies. The normal cell division for meiosis 1 on the left shows that the sister chromatids have not separated. We have 2 red colors in one nuclei and 2 green colors in the other. However, on the right, we see that the red and green are alternating in each of the 2 nuclei. This indicates that the sister chromatids were not together, and they did not monoorient. And this occurred with a frequency of 70% for our meiosis 1 data. So why are the sister chromatids separating prematurely at meiosis 1? Well, CG30383 might have similar functions to that of monopollen in yeast. The monopollen complex keeps sister chromatids together, and when it's mutated, the sisters segregate randomly. And this frequency of non-disjunction was similar to that in our experiments. Monopollen has not been discovered in flies. So this gene could be the fly's way of making sure the chromatids separate at meiosis 2 and not meiosis 1. Lastly, I'd like to thank my lab partner, Sarah Pellizzari, for all her help with the experiments and collecting the data. And I'd like to thank Dr. Tomkiel for all his help, support, and honestly for everything, because I would not have been able to do this project without him. Thank you.